the genomic marker location shown here. This line represents the genome-wide significance level of alpha equals 0.05. And you can see that there are 3 regions on 3 different chromosomes that influence the trait in this scan. The overdominance model predicts that plasticity will be a function of heterozygosity. And so I will calculate heterozygosity as the proportion of heterozygous loci in each individual and use linear mixed models to find evidence for or against a relationship. The allelic sensitivity model predicts that QTL will co-localize, that plasticity QTL will co-localize with trait QTL. And the epistasis model predicts that plasticity QTL will not co-localize with trait QTL. So to differentiate between these 2 hypotheses, I will use the trait values in cool and warm environments, as well as the plasticity value, and scan those for QTLs that influence multiple traits. To answer my 3rd question, what are the genetic correlations between the thermoplasticities of different traits? My hypotheses are that I can find positive, negative, or no correlations. And I'll test these hypotheses by taking the plasticity values of all the different traits and scanning those for QTLs that influence multiple traits. So now on to the broader impacts of this research. I will investigate the adaptive significance of thermoplasticity in natural populations and identify plasticity QTL for many reproductive and vegetative traits. This will give me the data to provide insight into the importance of those 3 genetic models of plasticity. My results will be useful for forecasting the response of organisms to global warming, and specifically will be able to inform crop breeders in preparing cultivars for future environments. So here's a timeline of my approach. Today is right here. I should be able to complete my F1 pollinations and produce all my F2s by this November at the latest. And then due to growth chamber limitations, I'm going to plant 2 cohorts of my family and go through and measure the phenotypes and then finish that up by the end of next year. And during this time, I'll be completing my AFLP analyses and publishing those results. I would like to thank my advisor, Dr. Elizabeth Lacey, for her unlimited supply of positive energy, as well as my committee members for helpful discussions, the Lacey lab members, both past and present, as well as NSF and the UNCG Biology Department for funding. And with that, I'll take any questions.
I'm just curious how you go about measuring pollen viability. Is that something you can look at under a microscope, or you
Yep,
have to
exactly.
actually?
Basically, you just put the pollen in a stain, and viable pollen grains are stained, non-viable ones are not. So you just count them under a microscope.
Good. You don't have to grow a bunch of plants.
I'm already growing.
A bunch more.
Right.
But maybe could you explain how cytoplasmic inheritance can cause this evolutionary lag that you mentioned about compared to nuclear genes?
Basically, what we see is traits that are inherited through cytoplasmic genes. Selection can occur on them without a response occurring in that population. And then after the selection ends, a response will then occur. That's an example of the time lag. But traits that are controlled by nuclear inheritance respond to selection without a lag.
Are the genes known involved in the change in pigmentation of the spike?
Well, we know that those spikes differ in alternative oxidation concentration, which is one gene, and several different anthocyanins, and the anthocyanin pathway genes are known. But in Plantago, it hasn't been identified which alterations in the pathway.
So this may be a similar question, but what kinds of genes do you expect to find that regulate plasticity, and what other examples have been found in other species?
Really, as far as thermoplasticity, there have not been other examples found in other species.
Phenotypic plasticity.
Right. So it basically depends on which trait and which environment you're talking about. So we know that flowering time responds to temperature and light. And as far as which flowering time genes have evolved, it can influence how that organism responds to the different environmental temperatures. So, what's that?
I guess I'm looking for specific examples because I'm wondering, how do you know it worked in the end?
How do I know my experiment worked?
Well, you find some genes going to your analysis, and then how are you going to show that? Are you going to take the next step and show that they're involved in thermoplasticity? That's not, you didn't talk about anything like that. So.
Right. Really, my work won't get down to the specific gene level because I'm mapping QTLs, which are genomic regions.
So how will you have a sense that everything went? I mean, you'll identify genes, right? All these studies always do. But then how do you know if you're correct? Do you have any positive control in a sense? You know, is there a gene you expect to find?
I really won't be looking at specific genes. I'll be looking more at genomic regions, which could be
Contain
very
specific genes.
Yeah. So as far as flowering time, we know the FLC gene is very important in the response to temperature and light. However, I'm not exactly sure of which specific genes I'm looking for at this point.
I take it the genome isn't sequenced.
Right, the genome is not sequenced, so
Correct.
you'll not know what kind of genes are in those QTL regions. But it's not guaranteed that a study like that will always find all genes
Well, not
in
all
our
of those, but
QTLs.
what I'm looking for is how do you know that how do you know it worked if you have no way of asking is.
Okay, so what I'm going to do is go through and identify QTL regions of the genome that influence the trait. This will be the first step if we are to go and do fine mapping and identify specific genes later. What we would do is take that QTL region and split it up into more experimental families and try to specifically see smaller and smaller regions until we can get it down to the gene level. How is the trait influenced by this piece of DNA?
Maybe you could talk about how you decide where a QTL is. I mean, what is your reasoning, or how is the logic, the scientific logic behind that?
Okay, so I'm having the genomic region in the same markers in my parents' F1s and F2s. And I also have their phenotypes. So what I'm doing is looking for statistical associations that show that individuals with this piece of DNA have this phenotype. And by using a genome-wide significance level of about 0.05, it produces an odds of about 1,000 to 1 in support of a QTL influencing that trait. But I will not get down to finding a gene unless by chance there's some of these traits influenced by 1 or 2 genes. But we don't really expect that because most of the traits that I'm looking at are quantitative.
No, I realized I was actually saying if you did know this gene involved in flowering,
Right.
and you know the general region of the genome, did that region come up as a positive or would you have that sort of some indication that you're seeing?
It's very unlikely because Plantago lanceolata doesn't have a sequenced genome, so I would have no reference to go back and find genes in.
So no genes have been mapped at all?